Greetings, my name is Kevin, and welcome to our Gene Editing Lab. Today, we have been given the opportunity to show you what gene editing is, how it works, and why it is important. We will also discuss two companies that we have worked with, and how they differ in gene editing operations. Let's go! Here are my colleagues, Ashley and Adrian. They will tell you about the basics of gene editing.
Hi there, as Kevin said, my name is Ashley.
And I'm Adrian.
And we will be going over gene editing and just how amazing this technology really is. First off, what is a gene? Well, a gene is a basic unit of heredity that is both physical and functional. It is required to create the structural proteins and enzymes necessary to reform and regulate tissues and organs. Made up of strands of genetic code consisting of guanine, cytosine, thymine, and adenine molecules, they make up all of our DNA.
From this, you can probably guess what gene editing is. Gene editing is a method that can rewrite the genetic code of DNA of all sorts of living organisms, resulting in many different useful changes, which we'll get into in a second. But how does this actually work? Basically, enzymes that target a specific DNA sequence in cells will cut into DNA strands, remove the existing DNA, and insert new DNA to alter your genetic code. Our other colleagues will go into further detail later, as there are a few different gene editing procedures.
Now, let's talk about how gene editing can be useful. The most obvious reason would be that gene editing can provide any living organism, such as plants, animals, and humans, with good genetics. I'm sure that everyone has heard of genetically modified organisms, or GMOs, which are often applied to agricultural crops. Currently, we create GMOs by adding genes from other organisms. However, with gene editing technology, we can produce even better crops by modifying the genetics to precisely create ideal fruits and vegetables. These modifications could result in larger and more nutritious crops.
As for humans, there are a lot of things that make us far from perfect. However, with gene editing, we can potentially reduce this gap through many different physical and mental alterations in the near future. For one, we can create the ideal human body type. By increasing protein and hormone production in cells, we can increase muscle growth and enhance athletic performance. To further improve ourselves, we can also genetically edit eye, skin, and hair color, along with other facial features. While these can be considered some nice aspects of gene editing, the most important reason for gene editing lies in the ability to cure all sorts of diseases and disorders. Many experiments have already been done, in which gene editing is able to modify immune cells to fight diseases such as cancer and HIV. And more recently, a study has been done at Stanford University which removed the genetic material of COVID-19, preventing the virus from acting on the human lungs. This case has proven to show that the removal of genetic material behind the virus has reduced COVID-19 in human cells by 90%. Not only can we modify disease risk, but we can also modify physical and mental disorders. There have been various promising tests which can potentially remove the genetic code that results in various disorders such as cystic fibrosis, sickle cell anemia, muscular dystrophy, and thalassemia. The final major thing that gene editing can also do is to alter the genetic code of non-human organs, specifically pig organs, in order to safely transport them into humans. This will prove useful for important surgeries, as organs are extremely valuable. Once perfected, this will greatly reduce the major costs and challenges of organ transplants.
Clearly, the benefits that gene editing can provide for humanity seems to be endless, as reflected through the many investors and companies beginning to pile into this rapidly growing industry. So now, we'll be passing it on to the representative of CRISPR Therapeutics, Janice, to talk a bit about how their gene editing technology works.
Hi, I'm Janice. Our research is on CRISPR Therapeutics, which is a certain gene editing company that, like the name suggests, focuses on using CRISPR as their main gene editing technology or method. The goal of CRISPR Therapeutics is to treat certain genetic diseases by using Cas9 proteins to turn off and/or fix unhealthy genes to their correct working versions. This can also be a treatment for cancer since Cas9 can turn off the gene that causes the cell to divide uncontrollably. Their gene editing process is based on using prokaryotic bacteria like the Streptococcus pyogenes, which can be found in the immune system. This bacteria would take samples of DNA from occupying viruses and would store them in a special part of their genome. Then, the viral DNA that was previously stored is fed into the Cas9 protein through RNA transcriptase. The RNA transcriptase then unwinds the sections of the viral DNA and builds an RNA molecule matching the viral sequence. Cas9 then binds to the section of the invading viral DNA to search for a strand that matches what the RNA had sequenced in. Once a match is found, Cas9 cuts out the viral DNA. The DNA will try to repair itself. However, it will most likely not be the same as before, making this useless. This process is used to turn off faulty genes, as any RNA sequence can be fed into the Cas9 protein. Additionally, synthetic DNA can also be injected as an attempt to repair the missing section from when we remove the viral DNA. Joyce, the representative of
Selectus,
will be talking to you about how their company works.
Hi, I'm Joyce. I'm going to introduce Selectus. Selectus uses talent as their gene editing mechanism. The goal of Selectus is to develop CAR-T therapies to treat cancer. Then, what's a CAR-T? CAR-T is cancer treatment that engineers the genetics within the body's immune cells to make them attack cancer cells. To engineer the cell for attacking cancer cells, they use talent gene editing, which I am going to explain to you. Talent is short form of transcription activator-like effector nuclease. Talent is a protein-based DNA targeting system consisting of a transcription activator-like tall protein and nuclease named FOK1. There are 34 repeats in the tall DNA binding domain, which can be differed by 2 amino acids. The amino acids determine which nucleotides to bind with. After binding with the target sequence, dimerized talent cuts the double-stranded DNA to fix it. From now on, I'm going to explain how CAR-T cells are produced. CAR-T cells are made by the method called knocking out and knocking in. Knocking out means disrupting the part of the gene to lose its function. Knocking out is for removing the possibility of graft versus host disease, which is when the donated T cell recognizes the treated patient cells as foreign and
attacks
the patient cell, which we want to avoid. Knocking in means insertion of external DNA at the cut site. At the cut site of the CAR cell, which is cut by the talent mechanism, the human immune cell T cell is inserted. CAR means chimeric antigen receptor. By combining those two cells, T cell and CAR cell, it can target and kill cancer cells. This is how Selectus is aiming to cure cancer using the talent system. Now, Edward will tell you about the comparisons between those two companies, CRISPR Therapeutics and Selectus.
Hello, I'm Edward, and I'm going to be talking about the comparison between the two companies, CRISPR Therapeutics and Selectus. Although both CRISPR Therapeutics and Selectus are gene editing companies, they have different gene editing processes. As the name suggests, CRISPR Therapeutics uses CRISPR as their gene editing method, whereas Selectus uses talent as their method. CRISPR Therapeutics uses CRISPR-Cas9 protein to develop gene-based medicines for many diseases. In addition to creating medicines, CRISPR has also been able to test and use Cas9 as a cancer treatment by turning off the error gene. On the other hand, Selectus uses CAR-T therapies for cancer treatments by making the body immune cells attack specific types of cancer. Although both CRISPR and talent have been proven to be effective methods for gene editing, talent has shown to be more efficient when compared to CRISPR, as it cuts DNA and edits the gene more proficiently. Now, I'll be passing it on to Kevin to wrap it all up.
Thanks, Edward. As you have just seen, gene editing has many useful advantages, such as creating genetically modified organisms, preventing and stopping viruses like COVID-19, treating different types of cancer, and many more. Companies like CRISPR Therapeutics and Selectus use gene editing, though they each use it for a different purpose. For one, CRISPR Therapeutics uses Cas9 to treat certain genetic diseases and cancer by turning off or fixing the incorrect gene. On the other hand, Selectus uses CAR-T therapies as a cancer treatment that engineers the genetics within the body's immune cells to make them attack specific types of cancer. Clearly, there are all kinds of gene editing techniques. We only went over 2 major ones, and there are even more uses for gene editing. As time passes, we will surely see gene editing become a common procedure in many various treatments and surgeries, and it will only get better over time. On behalf of the research team, thank you so much for listening and have a wonderful day.